Hello, everyone. Thank you very much for giving me the opportunity to talk about my study. I had been in Prague twice, and it is one of my favorite cities in the world. First, I'd like to briefly introduce myself. I'm a biology lecturer for the first year student at Mie University. After graduating from my first university, I worked for a company and stayed in Russia, both in Moscow and in Kamchatka. During my time in Kamchatka, I noticed that some plants, such as Artemisia, Sausarea, and others, looked similar to those found in Japan. So after returning to Japan, I enrolled in a second university to study plant taxonomy. At the university herbarium, I compared several plant specimens collected from Japan to Kamchatka. I found that Micranthes fusca would be interesting subject to study because its morphology changes abruptly in the middle of the Kuril Islands. Micranthes fusca is usually a delicate plant with an elongated inflorescence and sparse flowers. However, on Itrup Island, it suddenly becomes larger with a broader and denser inflorescence. Later, I began to suspect that Micranthes nelsoniana might be related to this morphological change, since Micranthes nelsoniana is commonly found from the central Kuril Islands northward. The map shows the distribution of Micranthes nelsoniana and related species. Micranthes nelsoniana is a large species complex that includes 7 to 8 varieties, mainly distinguished by plant size, the presence of hairs on the inflorescence and leaves, inflorescence shape, and other characteristics, although the boundaries between these varieties are unclear. It usually grows along streams and rivers, but can also be found on mountain slopes, coastal rocks, and occasionally in meadows. The species also known for its high ploidy level, which has been extensively reported from Chukotka, north of Kamchatka. Micranthes fusca is distributed in Japan and the southern Kuril Islands. The species is characterized by its thin, elongated inflorescence and distinctive flower shape. The petals are dark red or pale green. The filaments are short, less than half of the petal length. And the gynoecium remains short during flowering. It blooms in late summer from August to September. Micranthes ovi is endemic to Kamchatka, where it grows on volcanic slopes. It also occurs in the northern Kuril Islands. This species is characterized by its red flowers, thick and shiny leaves, and its specific habitat. Its floral structure is similar to that of Micranthes nelsoniana. It is interesting that these species, along with other closely related species of Nelsoniana, are found only in this region, the southern part of Northeast Asia. So the aim of this study is understand the genetic relationship between Nelsoniana, Fusca, and Owi in Far East Asia, and study chromosome numbers and ploidy level around this region. We mainly collected plant samples in Far East Asia, For Micranthes fusca from southern Kuril Islands, I had an opportunity to visit through a visa-free exchange program to the Northern Territories and with the support of the Kuril Nature Reserve. Phylogenetic analyses were conducted based on the ribosomal DNA ITS region and combined globulus DNA region. Chromosome counts were also performed. Samples of Micranthes nelsoniana from Hokkaido, which have high chromosome numbers, were cloned to analyze their ITS sequence contents. Here are the sampling localities. Dots of each color indicate the exact localities. Localities marked with circles represent samples whose ITS sequences were obtained from DDBJ. Samples from Japan to Kamchatka were collected intensively. This is the result of ITS phylogeny. Micranthes nelsoniana and related species form a large clade distinct from outgroups. Within this clade, we identified a distinct subclade consisting of nelsoniana, fusca, and owi, and others. The ITS phylogeny was compared with the geographical distribution. The large clade, excluding the subclade, mainly consisted of Micranthes nelsoniana, which covers a wide distribution area from Siberia to Alaska. Within the subclade, two clades corresponding to Micranthes fusca, one from the southern Kuril Islands and one from Japan. The next clade corresponds to Micranthes manchuriensis from Primorye. The next clade corresponds to population of Micranthes nelsoniana and oi from Kamchatka, as well as Micranthes nelsoniana from Hokkaido. Finally, the last clade includes three species: Micranthes nelsoniana, fusca, and owi, occurring from the central Kuril Islands to the Aleutian Islands. The results indicated that the marginal island regions of Northeast Asia, including the Kuril and Aleutian Islands, the Japanese archipelago, and Primorye, have a unique genetic groups compared to the continental populations of Micranthes nelsoniana. The ITS phylogenic tree was compared to the chloroplast DNA phylogenetic tree. Within Micranthes fusca, the two phylogenetic trees corresponded to each other to some extent. However, the remaining parts of the trees were more complex, with some samples corresponding to different species, suggesting active gene flow not only within species but also among different species. The ITS haplotype network was constructed. This section represents continental part of Nelsoniana. This section includes Micranthes nelsoniana and owi from Kamchatka and Hokkaido. And initially, I focused on Micranthes fusca. The red circles indicate Micranthes fusca. In the groups from Japan and southern Kurils, all the plants with these haplotypes were Micranthes fusca without exception. However, the circles within the green squares included three species found from Iturup Island in the Kurils to the north. The red segments within these circles corresponding to Micranthes fusca, mainly with broad, dense inflorescence. This is a chloroplast DNA haplotype network. As in the IDS network, the red circles on the left corresponded to migrant fusca, and the red segments within the green ranges represent hybrid fusca individuals north from Etrup Island. Within the network that includes continental Micranthes nelsoniana, we identified two separate parts of the green range, suggesting the presence of at least two distinct chloroplast DNA haplotype lineages for hybrid fusca. The real Micranthes fusca has short filaments styles, petals with reflected margins, flowers that bloom in August, September, and grows along rivers. Among the plants collected, we found only one example of hybrid type 3, which has the same haplotype as the real Micranthes fusca, but its IDS type was within the green range. The hybrid appears similar to the real Micranthes fusca in terms of inflorescence with sparse flowers, habitat, and flowering time. The rest of the plants were all type 4, where both parents are included to the green range. These plants had petals with a reflection, larger capsule size, and grew in various habitats such as coastal areas and meadows. This suggests a tendency for introgressive hybridization from nelsoniana to fusca. Another interesting aspect of this region is the distinct distribution of plants within the same clade, one in Kamchatka and the other in Hokkaido, Japan. In Kamchatka, Micranthes nelsoniana occurs at elevation of 1,300 meters. Micranthes oi occur at 1,300 meters. In Hokkaido, Micranthes nelsoniana occurs at 1,700 to 2,000 meters altitude. The distinction in distribution may be related to the high elevation habitats in which these plants grow. Herbarium specimens indicate that Micranthes nelsoniana from high elevation also occur at 1,500 meters on Etrup Island, although we have not yet collected these samples. Chromosome numbers in these regions were studied. Since 2n equal 28 is commonly observed in nelsoniana, it may represent the basic number. The chromosome number Micranthes nelsoniana increased up to 2n equal 80 or 84, particularly around the Bering Strait. The chromosome number of Micranthes fusca is typically 2n equal 30. In the Kuril Islands, 24 and 50 are commonly observed. Micranthes nelsoniana in the northern Kuril and Micranthes oi in Kamchatka both had some of them both had a chromosome number of 24, and some of them shared a close plus haplotypes, suggesting possible past hybridization. It's a little bit reddish. The presence of the same chromosome numbers may facilitate gene flow in this region. Chromosome studies revealed that Micranthes nelsoniana from the highlands of Hokkaido has large chromosome numbers. One of them 50 and the other is 80, which correspond to the tetraploid and hexaploidy, respectively. Therefore, a cloning experiment was conducted to determine the genetic composition of the IDS region. The samples from the highlands of Kamchatka and Hokkaido, which had two different chromosome numbers, all belong to the same pink clade. A plant with the chromosome number 50 from Hokkaido also had the sequence that didn't belong to any clade of nelsoniana, here and here. A plant with chromosome number 80, in addition to them, had a sequence from the green clade, where three species are mixed in the lowlands of the Aleutian Islands. These results suggest that the plants from Hokkaido contain sequences from both ancient lineage and island lineages, indicating that Hokkaido has been involved in active genetic exchange within this region. Conclusion: unique genetic groups comprising Micranthes nelsoniana and closely related species are found along the marginal island regions in Northeast Asia. Micranthes nelsoniana makes contact with Micranthes fusca in the central part of the Kuril Islands, resulting in several types of hybrid plants. Some of these hybrids shared the same chromosome numbers, which appear to promote hybridization. Highland plants of Hokkaido contain sequences from both ancient and island lineages, suggesting active genetic exchange in this region. Our study revealed a significant role of the marginal islands of Northeast Asia in the genetic diversity of Micranthes nelsoniana and related species. Thank you very much for your attention.